Today, I will be discussing different types of point mutations and their effects on proteins. Here we are given a DNA sequence in vivo. The top strand reads 5' to 3' and the bottom strand reads 3' to 5'. A protein that is 4 amino acids long is formed from this DNA sequence, but in order for this process to begin, the DNA has to be transcribed into RNA. To help determine which strand is the template strand and which is the coding strand, I have flipped the bottom strand so that we can read both strands in the same direction. By doing this, we can see that both strands have a start codon, AUG, because the T becomes a U in the mRNA transcript. This strand, though, is the only one with a stop codon, which is 4 codons down from the start codon like it is supposed to be. After switching this A to a U, we have UAA, which is a stop codon and does not code for an amino acid. So this here is our DNA coding strand. Now that we have our DNA coding strand, we can transcribe our mRNA sequence, which is done by changing T's to U's, like shown here in the wild-type example. 4 codons starting with AUG to AAG will be translated to form the 4 amino acids shown here. Each codon has been color-coded to help you visualize the different codons and their respective amino acids they code for. If a mutation were to occur that changed the guanine that was here to an adenine in our DNA coding strand, the same would occur in our mRNA sequence. This mutation would not result in a change in our amino acid sequence, though, because AAA and AAG both code for the same amino acid, lysine. This is an example of a silent mutation because a swapping of a nucleotide base pair does occur. However, no effect is present on the amino acid sequence. The substitution of G with A seen here is an example of transition since both are purines. If a transversion were to occur that swapped the same guanine for a cytosine, a change in the amino acid sequence would occur because AAC codes for asparagine and not lysine. This is an example of a missense mutation because changing one nucleotide base causes an alteration of the amino acid sequence. This mutation can affect the function of the protein depending on whether or not the affected amino acid is vital to the protein's function. Looking at another example, if a transversion were to occur that changed an adenine to a thymine, a UAG codon would be translated on the mRNA after transcription occurs, instead of the AAG seen on the wild type. This means the codon that used to code for lysine would now code for a stop codon that will end translation. This is an example of a nonsense mutation, since a single nucleotide change leads to a shortened amino acid sequence. This would likely result in a non-functional protein. In the last example, we have a transversion here on the DNA strand that would cause a UUA codon to be translated instead of a UAA codon in the wild type. In the wild type example, the UAA is a stop codon and stops translation, but UUA codes for leucine, so the stop codon is not present, meaning the mRNA will continue to be translated until there is another stop codon in the reading frame. This is an example of a read-through mutation and would result in a long, non-functional protein. Here are the external sources I used for this video.